To get started, I'll just introduce myself. My name is Emma. I know that you guys haven't had me before yet. I am just going to be teaching this week's lecture, which is like molecular biology focused, and then next week's lecture, which is more cell bio and kind of components of the cell, which is what I really like. A little bit about me. I went to Cal for undergrad, so go Bears if anyone's Berkeley out there. I studied integrative biology. I then did Teach for America and taught 7th grade biology. Then I went back to school. I got my master's in biomedical science, which was incredible. And I'm heading to medical school in January in Australia, which I'm super excited about. And thanks. And yeah, if anyone has any questions about MCAT in general, applications to medical school, international applications, I'm happy to answer any questions. You'll see on the slides that my contact info is there. Feel free to reach out if you have any questions. I think this lecture, fingers crossed, should only be about an hour. So we'll definitely have time at the end, or maybe if you're running off to something else, it'll be nice to have a little extra hour. So, and I took this class, so I know that timing can be a pain when it goes over. So I definitely am not going to try to go over. We'll stop no matter what at 4, but hopefully we'll be done early. So I see already a few messages in the chat about kind of medical school. I maybe will touch on those kind of at the end, just so we can dive in, get started, hopefully get wrapped up early, and go from there, if that sounds okay. I am going to lecture off of my iPad. So I'll share those slides and I'm going to turn off my video. Not because I don't think you'll enjoy watching me, but basically because I just talk a lot with my hands and it'll be less distracting. So let me send the slides again so everyone has them, and then we're going to dive in. There are the slides again. And if someone doesn't mind, if anybody else joins late, just reshare them or direct message them with those slides. So let me get started and share my iPad over here. All right, I can see that on my end, but if just let me know in the chat if there's any kind of issues going on if you cannot see those slides. So as I mentioned, this is the molecular biology lecture. My office hours are Tuesdays, 9 to 11. And that's my email there. You can feel free to email me anytime. That's my personal email. I don't know if, yeah, Todd has shared it, but if you are looking for any like private tutoring, I'm also happy to help that. That is California time, Pacific, but if you need to meet outside of those hours, just shoot me an email. I'm actually relatively flexible right now. We can definitely set up time. And if you're interested in like private tutoring as well, just send me a message. We can definitely chat about like what might seem like a feasible option for you. So let's kind of dive in. This is the AAMC topic list for molecular biology. This is, I would say this is kind of like a weird group of topics is the word I want. Sorry for that. And thanks, Charu. And they're kind of, I think the most important thing to focus on is kind of this last point here in the practical applications. Everything else you really need to know at kind of like a high level perspective. So we're kind of just going to gloss over a few things, not gloss over, but like go over each topic and kind of get the basics. Definitely feel free to ask questions. I've got the chat open, although I will tell you it's sometimes a lot of questions go by. So if you feel like your question doesn't get answered, hold it for the end or just feel free to resend it. That is definitely totally fine. So we're just going to dive right in. So you might have heard of gene cloning before, as it kind of makes sense. It's about cloning a gene or making copies of a certain gene. So we might want to do this because a gene is shorter, obviously, than a whole set of DNA or an entire set of chromosomes. And we might be looking to study the different effects of the gene or what happens if we knock out that gene, and just kind of different things. We often are going to use a plasmid, basically to insert our gene onto, which is just kind of like a generic segment of DNA. And then we often will force a bacteria to kind of take up that DNA. Plasmid and vector are often the same thing. So I wouldn't worry too much about like what they are. You're basically, you can see in the next slide, we're going to go on to plasmid DNA, which is often called vector DNA. That is kind of just like our carrier. Oftentimes we are going to then produce this recombinant DNA, which is basically our plasmid or our vector with our gene. So they've been recombined into two things. And then we're basically going to force a bacteria to take that up and then see what happens. The orientation does matter. And this is something that you might see on the MCAT, where we've got kind of our gene and our recombinant DNA. And we want to make sure that when we're inserting our gene, we're following the same kind of order or direction of the way that we want this to be transcribed. If we had our recombinant DNA here and we inserted our gene of interest here, but the gene is actually facing this way and our order of replication or our direction of replication is going the other way, we're going to run into issues here because we're transcribing this way and suddenly our gene is backwards. So potentially that's going to give us a different outcome. So it's really important that we're following directions. So if you ever see questions about that, just double check the direction, make sure everything's lined up correctly. And this is also important because basically when we're doing this whole process, we're trying to like hack into a bacteria or hack into whatever cell type we're in to basically take over that host machinery and produce something. So we want that host to think this is as normal as possible. And so therefore we want to make sure that we're choosing a really similar direction to the way that the current kind of DNA flow is going or the way that it's going to be read. So sometimes you can look for promoter regions and like they'll highlight that and be like, oh, this is the promoter right here. And so P for promoter. And so we know that maybe it's going to be read this way. So we can tell that this would be an incorrect orientation. I'll just note that in case anyone like takes a screenshot of it. So this would be incorrect because we've got competing directions here. We want to make sure everything is going in the same direction. So in order to do this kind of process here, where we're like cutting out genes and inserting genes, we're going to use something called restriction enzyme. And a lot of this might be review. Michaela just asked, promoters encourage the translation of the gene. Potentially, I don't know if you have had the like lac operon lecture yet, or kind of like gene transcription and things like that. Promoters are basically sections that potentially are going to help identify where the gene that we're trying to transcribe is, or potentially if they are modified in some way, they may like turn on or turn off the transcription of that area. But they often are like kind of have like little signals similar to our restriction enzymes. They're signals that they're looking for in order to bind different enzymes there and could impact either positively or negatively the transcription of that, translation of that area. So when we jump into restriction enzymes, which in this case are going to help us, they are enzymes as they've gotten their names. They are going to be an endonuclease. You probably had the lecture where you talked about all the different enzymes and all like transcription, translation, all the different ones that are in there. And so an endonuclease is going to cut the bond in between two nucleotides. And this is important because we're basically trying to extract our little gene segment from the DNA that we're interested in. So they are going to recognize specific sequences of bases, often called a recognition sequence. And this is kind of, when we're thinking about this, we don't just want restriction enzymes floating around all the time, thinking about, where they can cut, because then all of our DNA would be cut up. So it's really important that self DNA can be protected, similar to like DNA being protected any other time. It can be methylated. There are histones involved. Sometimes the nucleus is more or less permeable. So we know that we can protect our own DNA or DNA that we're not interested in looking at. And we can just look at these restriction enzymes. We'll just look at genes or areas that are exposed that are not necessarily doing that. And restriction sequences are often palindromic, similar to like the TATA box in a promoter that's palindromic. So I've definitely seen some questions asking you like, what's the most likely restriction sequence, giving you a bunch of like nucleotide sequences. And then essentially you're looking for ones that have palindromes, either at the beginning or the end. The whole sequence doesn't need to be a palindrome, but often you'll see that the beginning and the ending are palindromic, which just means that they are the same forwards as they are backwards. And sometimes when these restriction enzymes cut, they produce what are called sticky ends. So you'll get kind of like, if this is my one side and my second side, when they cut in here, or actually what I'll do is I'll go like this. So my restriction enzyme is going to come and latch on. It's then going to know that it needs to make a cut. And so it might actually make a cut here and a cut here and a cut here. Oh, sorry, that's the wrong way. And a cut here and a cut here, hypothetically. And then I'm going to be left with a shorter end and a longer end of both what it just came from. But I'm also going to end up in my little piece that's being cut out, right? This is actually a little bit, they're not quite overlapping. In my little piece that's cut out, they're also going to have like these offset ends. So when sometimes you see the code or sometimes they'll like write the nucleotide bases, you'll see kind of these areas at the end here that are overhanging. And there's not really like a necessary reason why they do or don't do this. Some of them make blunt cuts, which just means that it's basically exactly the same. So if I had my section of DNA, my restriction enzyme might cut at exactly the same place. And I've just got an individual kind of like sequence cut out that's doesn't have those overhangs. These are just kind of important things. Sometimes they'll ask you to like predict what the complement is going to be. So that would be a case where they have kind of those sticky ends that you could actually predict what the other side is going to look like. So there are 3 types of restriction enzymes. I wouldn't worry too much about knowing exactly what restriction enzyme does what, but I think it's good to kind of refresh yourself and remind yourself that there are 3 different types. Type 1, they have known recognition sequences. They know where they're supposed to cut. However, they actually make a random cut within 1,000 base pairs. So you're going to have a lot of variation there. Type 2, they're going to cut exactly at the recognition sequence. And type 3, they also know their recognition sequence, but they cut within 25 base pairs. Oh, sorry, I left off base pairs on the end there. So you can kind of see there's a difference. There's not really like a reason to know why there's a difference, but sometimes they'll ask you, like, oh, this is a type 2 restriction enzyme, and here's the example. And so you don't need to know that it cuts exactly at the recognition sequence because it'll kind of be shown within the example, if that made any sense. Someone asked, what does it mean if they cut within a certain number of base pairs? So let's say I'm just going to choose. Okay, so this would be my recognition sequence. So my enzyme is going to come along. Sorry that I've decided that this is the enzyme. It's going to recognize this, but then it's actually potentially going to move either 25 base pairs this way or 25 base pairs this way, and then decide to make its cut. So if you were thinking about, I know that sometimes you get asked like theoretical, like experimental design questions within the MCAT. So this could be a reason when they say like, okay, these are the 3 types of restriction enzymes. Which would you choose if you wanted to check exactly a known region of a gene? You might pick type 2. But if it says, if you're not really sure where the gene is, what might you use? Or if you know that the gene is within this chromosome between this base pair and this base pair, which might you use? And so those are times when it can be come into play, like the differences of using potentially a type 1 versus a type 3. So that cut can happen anywhere between either 1,000 or 25, depending on which type it is, base pairs of the recognition sequence. So you may or may not miss the gene with that kind of cut, but oftentimes we're doing this in a lab kind of setting. So we're either aware of where we're cutting or aware of which restriction enzyme we're using. So the process can kind of be simplified by this. And I think what's important to remember here is that you're really thinking about like big picture things. Don't get caught up in the details of things. You could find, of course, online like 50 different ways that this process might go on. But this is kind of the overview process of a restriction enzyme. So the first thing is that we're going to find. We're looking, and I kind of like simplified this in like a Word document type thing. So we're finding, and so we're looking for the recognition sequence. That's when that comes into play. Remember that we're thinking of palindromes, which are the same forward and back. Once we've found our palindrome, we're going to actually cut. So that's where our restriction enzyme comes in. You might have seen them as like EcoRI, which is just like another one. I think there's one that's like starts with an S. You don't really need to know those weird names. They often are named for like whatever bacteria they're with or from. Sometimes they're named about what gene they're working on, but they will often give you, they're not going to ask you like which restriction enzyme is more likely. They're just named just like every other enzyme. But we do remember that they are going to be an endonuclease because they're cutting in between those nucleotides. Then we're thinking of our paste step. We're usually pasting this into the plasmid, which is what we saw earlier when we had our plasmid, and then we had our little gene that we're going to insert in here. And then basically to attach this, just like when we were attaching any other kind of DNA strands together, we're going to use DNA ligase. So that's like a really good thing to remember. Ligase is coming in again, and we're basically going to try to connect those little sticky ends or maybe blunt ends, whatever they are. So then this is considered your recombinant DNA altogether, this whole thing. And we're trying to see what's going to happen when this gene is maybe expressed or maybe suppressed. So then we're going to mix it often with a bacteria. They're really simple. They reproduce quickly. And so I don't know if you've done this in at Cal. I think we did it in Bio 1B lab, where basically you force the bacteria to uptake your new plasmid that you've created, or recombinant DNA. Often with heat, sometimes with agitation, stirring it. I think we put ours in a blender. And then basically you're going to do the copy part, which means you're going to grow it on a plate. Often you have nutrients, so it's growing quickly and, thriving in its little environment. And sometimes with antibiotics, this is actually how we find out whether different genes maybe make an antibiotic more or less resistant. So sometimes they're grown in the presence of antibiotics, or you co-insert a gene for antibiotic resistance to see whether that gene changes anything. So a lot of times we're using this in like a very theoretical lab-based scenario, this kind of like recombinant DNA technology. It's not necessarily happening a ton within our own cells, which is totally fine. This is how we like learn about stuff. And I'm pretty sure this is actually the way that even like some pharmaceutical drugs are made, like insulin. So it's quite cool. And somebody asked if we're always looking for a palindrome. I want to say almost always. It's kind of like because it's that recognition sequence, it does need to be like uniquely identifiable. And so kind of those palindrome sequences help with that, so that it can potentially view it or like recognize it in either direction, which is what's helpful about a palindrome. I left that out. All right, so we're going to move on to DNA libraries. I've got another question here. So any recognition sequence is just a palindromic area that basically the restriction enzyme knows to recognize. It can be a TATA box. That's like a one for DNA replication. It doesn't have to be though. Sometimes like a TATA box is part of a promoter region, but just any palindrome. So it could be basically any sequence of of nucleotides that are going to be palindromic. So it could be like GC, 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 GC. It could be GGGC, GGG. That's a palindrome. It can be any palindrome. And I don't think they're going to like ask you to like prove one. There aren't like known ones or unknown ones, not like codons or anything like that. So it's more just like if you had, or here, we'll go back here. So we'll say, which of the following is not a recognition sequence? I'm just gonna, sorry, my handwriting is not super clear. All right, so this is gonna be A, B. You can type in the box which one you think it's not gonna be. Which of the following is not a recognition sequence? Yeah, A. Because here I've got AA. Do I have AA at the other side? No. So automatically no. Excellent, Sophie. If we added though, like an AA here, we're still not at it because here we're going, this side we go AAGG. Here we have AACC. So we're still not going to be here. With the second one, AATTCC, AATTCC. So those work. That's a palindrome. GCCG, that's a palindrome. Same going forwards and backwards. So it just helps for that restriction enzyme basically to know where to bind. So moving on to DNA libraries. Now we're thinking about like looking at just like different things. So DNA eventually can help us make proteins. I'll switch so we can have this little protein right here. And it might be an enzyme, it might be some other protein in the cell, and we might be thinking about how is this made? Well, we know in order to get this protein that we basically have taken DNA and we've turned it into mRNA to eventually get a protein. That's a super simplified stream kind of like what's going on. But we're starting now with the protein side of things, and we're trying to figure out what the DNA is that codes for that protein. So this process of having a protein, identifying it, and trying to figure out what the DNA is, this is what is called a DNA library. And this is not necessarily helpful to you and me on a regular day, but if you have worked in research or you know different things about research, you know that this process could be really helpful. The actual, being able to look at a protein and know what DNA actually codes exactly for that protein would be really helpful. So the limitation is though, when we're looking at proteins and we're trying to go backwards, so if we redraw our process here, in between proteins and mRNA is the amino acid sequence. And this is where we run into a little bit of problems because the amino acids, right, there are multiple codons for the same amino acid. So this is somehow, or at some point where we run into a problem, we can't easily go from protein to mRNA. We'd get many different types of mRNA or many different kind of variations. So we kind of make a little workaround for that, and that is called a cDNA library. So in the cDNA process, we are going to find the known mRNA. So we know that this mRNA codes for our little protein of choice, which I think was this guy. So we know that these two are friends, okay? We know that. From our mRNA, we are going to produce a single strand, hit my eraser, a single strand of DNA that is known as cDNA. The C stands for complement. Okay, and this is because this cDNA is actually complementary to the gene that we are looking for. And we're doing this via reverse transcriptase. So our transcriptase helped us to go from DNA to RNA when we're doing like all of our kind of like DNA replication and copying and making mRNA transcription. But now we're going to do that in reverse. So we know that we're going to get from RNA a single strand of DNA, which is called cDNA. And then in order to actually figure out what the whole DNA sequence is, we need to be able to turn this single-stranded DNA into double-stranded DNA. And so that is going to happen via DNA polymerase, which makes sense. Anytime we have damage in our DNA, we can basically help figure out what's going on via DNA polymerase. So these are sometimes just they're really important kind of enzymes to know what are the players in the game. And these are our two friends here. We're going to use reverse transcriptase and then DNA polymerase to get our double-stranded DNA. Once we have our double-stranded DNA, we could actually use a restriction enzyme to insert it into a cloning vector, have it be amplified, and then we'd get the sequence, and we'd be able to understand that sequence by looking at these base pairs. And there's lots of like technological things that you'll go over kind of in the lab techniques, which I think is sometime next week, lecture about how you can actually like look at those. This is kind of like a random side note, but if you're ever trying to remember if it talks about like northern blotting or southern blotting, you may have seen this, but this was something that I found really helpful, was something called snowdrop, which just kind of helps you identify who's going with who. So southern blotting, you're going to look at DNA. Northern, you're going to look at RNA. The O's are nothing because there's no like other. And Western is looking at proteins. So it might ask you like if you're using cDNA, like which type of lag technique would you do? Southern blotting, northern blotting, western blotting, none of the above. And if we know we're looking at DNA, then we know we're going to be in the southern region. If it's asking you to look at the mRNA, we'd be looking at northern. And if we're looking at our proteins, we're going to be doing western. So I'll just kind of go over briefly the differences between those two. So in our, we'll start with this one. So in our cDNA, right, we're actually going to make complement DNA. And that's because we take from this mRNA and we're going to produce a single-stranded DNA of what coded for that mRNA. So it's kind of like the steps in reverse. So we're going to go from our mRNA back to our DNA, but this is going to be a single strand of DNA, and that is going to be the complement to our double-stranded DNA. So this is the cDNA library really tells me from the mRNA to the DNA. And a cDNA library is always single-stranded. This is like what's really important. Compared to our DNA library, which is basically just identifying any protein and knowing the double-stranded DNA sequence for it. So if they ask you, we have a question coming up, which we'll practice it on, but if they asked you like, what's true about a DNA library? It might say like, DNA libraries are double-stranded DNA sequences. DNA libraries are made from RNA polymerase. DNA libraries are like describing what sequences of mRNA are. So those second options would be wrong because our DNA library is double-stranded DNA and it identifies the DNA sequence of proteins, the double-stranded DNA sequence of proteins, compared to the cDNA process, which is looking at the single-stranded sequence of mRNA molecules. So the DNA library, yes, Michaela, the cDNA means we meant the antisense, because remember our sense is really what we're reading and our antisense is like what the opposite is, which if you remember kind of in your like DNA, when you're looking at it, right? Here's your little unzipped region, when our enzyme is coming along to like read this, it's shooting out the RNA, right? That's going to read the sense strand, but it's going to be complement to that, which is eventually going to get the protein. But when we're looking at our mRNA and we're going back to our DNA, we're making that complement side. It's a little tricky. I know the words kind of get confusing. I would really highly recommend watching a video online just to kind of like really hammer that down and like visually see it. Sometimes that helped me if you're feeling like a little bit lost. But definitely you can ask me after and I can try to explain it in a different way as well. But some of those things that we said, so we said the cDNA library is going to be complementary. It's produced by reverse transcriptase from mRNA, and it's going to be single-stranded compared to if we made like a little comparison to our DNA library. Really, honestly, the thing about a DNA library is that it's kind of theoretical in a way, but it is going to, you're going to be able to identify the double-stranded. Identify the double-stranded sequence of a DNA protein. Okay, so those are kind of some comparisons between those two. Sorry if I'm talking a little bit fast. That is definitely something I'm working on. So we're going to go kind of onto our next like two topics, which are definitely going to be covered in the lab techniques lecture next week. So any more questions kind of on cDNA or DNA libraries before we jump in? All right. The next thing that kind of is like a topic in here, and sorry, this feels like very disjointed, but oh, sorry, Sophie, what does it say next to DNA? Sequence, SEQ, sorry. Whoop. We're going to, I was just saying like these kind of topics feel like really disjointed. They are kind of a little bit, I would say this is almost like a biotechniques, molecular biology type zone. If you are already taking practice tests, you might have seen that within kind of like the bio section, sure, there's like a lot of theory and a lot of other kind of like topics that are being covered. And then every once in a while, they kind of throw in these like experimental design, biotechniques, lab technique type questions. So they're not super common. It's not like it's 50% of the test, but they definitely pop up. So it's just nice to be familiar and, you know, kind of like review some of these different things, but don't kind of like get too overwhelmed by all of the little details. It's really beneficial just to have like a big kind of picture view. And we can definitely go and do kind of review. I also was talking to Josh about maybe doing some like general bio reviews. So definitely let me know if that would sound of interest. I know that kind of like putting all these pieces together, sometimes you can like really dive into like all of the like really nitty gritty. And it's really nice to just like have a refresh from the 10,000 foot view. So that would definitely be possible. And I'm happy to like put those together. So sounds like it's a positive kind of response from that. So great. I might ask at the end just to get a few more ideas so I can jot those down, but we'll kind of keep moving on into our next section here. All right. So the next two are kind of more like techniques, I guess, like lab techniques. One is called DNA hybridization. I really think this is like low importance, honestly. Feel free to disagree with me. But essentially, DNA hybridization, as it might sound, is like hybridizing or putting two different DNA strands together. And it's used really to compare similarities between DNA strands. The 10,000 foot view is you're going to denature double-stranded DNA from 2 samples, mix then your like single-stranded pieces. So if you had like this guy, oh, that's not really a DNA. This guy, sorry, these are like some of my worst DNA. And he is maybe semi-related to this guy. Wow, look how much better that one is. You're then going to separate them. So they're now all single strands. Do a little arrow. Sorry for that. And then once we have all our single strands, you're going to throw them in a test tube. What else do you do in biology but throw things in test tubes? And then you're basically going to see if they anneal. Sometimes you do that with like heat. Sometimes it's with other agitation. And you're going to see what is going to happen if I say, oh, plus Q, which can be like a shortcut for heat. How much of them are going to overlap? So is it going to be like this red guy and this purple guy overlap like this? So is this section my similar section, or are they just all going to be in there kind of like spaghetti still hanging around, which means they probably have little in similarity? That is it in like a nutshell. More locations of hydrogen bonding mean that they're more similar. Less locations of hydrogen bonding mean that they're less similar. That is like the whole thing. There are other ways that you can kind of like test this. A scenario where you might test this is you might say like, well, these two, like maybe they're like relatives is an option. Maybe it is a normal cell, what we consider normal cell versus a cancer cell. So you're looking to see how much of my DNA has changed. Maybe you're looking for, you're trying to figure out what is like the common section of DNA in 2 known ancestors. So you'd be looking for things like that. But those are examples of how you could use DNA hybridization and the very simple process of what DNA hybridization is. As I said before, I think this is low importance. There's not really like much meat here. Next would be a PCR reaction, which you've probably heard being talked about a lot. Probably many of you, if you're on campus this semester, have had many PCR tests. But PCR stands for polymerase chain reaction. And ideally, right, we're using polymerase. So we're definitely going to be building a lot of like a lot more like strands up. And it's a chain reaction, which means we know there's going to be a couple steps involved. I am not going to go like super in depth in this because it's going to be in the lab techniques lecture. And honestly, if you feel like even in the lab technique lecture, it's not really covered well, you can come back to me, you can find a video online, try like looking at some homeworks, but really, I don't think this is also going to be asked about in like really specific questions on the MCAT of like which enzyme did this. You just kind of need to know that 10,000 foot perspective. So ideally, you are going to have your little test tube. You're going to have your double-stranded DNA, which once again, my DNA is terrible. I'm sorry. Double-stranded DNA inside. We are going to add some heat. Q. And we are going to break those hydrogen bonds. So now we have, oh, I should do my test tube the same color. Now we have just like tons of little squiggly bits of our single strands. We're then going to add a bunch of primers, add a bunch of nucleotides. We're going to hope those things basically have a party, essentially. Very simply, they're going to build up. We're going to run DNA polymerase. We're going to make sure there's DNA polymerase in there. And now we're maybe going to end up with 2 times the number of strands. So we started with 2 strands. Sorry, I added an extra one in here to go accurate. So we had 4 strands of single, and now we're hoping to have 4 strands. Oh dear, you guys. Oh, all right. Slowly but surely, we're going to get this done. No, okay, ignore the L. we're just going to. Okay, so now we have 4 pieces of double-stranded. We then would repeat the process again. Add some heat. So now we have 8 pieces of single-stranded. And this is basically how, when you are looking for in a PCR test, right? You have to get the swab, you stick the swab up your nose, turn it around up there, feels like a brain biopsy, take it out, put it in that liquid. Basically, in that liquid is going to just be some preservation stuff, or just like basically like usually it's like a buffer solution just to kind of like keep it normal. It goes off to the lab. The lab is going to get that test tube, add some heat. It's going to split those double strands of DNA. Then once you're in your single strands, you're adding your primers, you're adding your nucleotides, you're going to rebuild your strands. You're going to do this multiple times. And then you are specifically looking for, I think they're still looking for the nucleocapsid. They might be looking for different things. The nucleocapsid of COVID, the spike protein of COVID, they're looking for anything that would signify that that viral DNA is present. And they can look for, even if they just say, just this one, the virus has inserted itself right here. Once I've copied it, right, and it's right here, now it's going to be here. And the next time it's going to be 4 times. Sorry again, until we're built up. So this is how PCR tests can be helpful and how polymerase chain reactions can be helpful to actually like identify if there's that small presence there. Now, I did put in here, hopefully, mostly because I was just kind of playing a joke. I don't know if any of you have had COVID. You may have tested negative when you had COVID. So it's not, it's not totally accurate. Sometimes you can get a bad sample. Sometimes it doesn't work. And it doesn't tell you whether that virus is alive or not. It just tells you whether it's present, with that section of DNA is present, or whether that virus maybe like has been in your body. So if any of you have had COVID and then you've continued to test positive, this is why, because it's still there. And this is why you can test positive for up to 90 days. Exactly right. So it is COVID testing, definitely super cool that we can use this really basic reaction to actually look for it. But it's not always super helpful. Just tells us whether it's present or not present in that. Let me go back and look at some questions. Yep, somebody asked, is PCR a method of DNA hybridization? It's not, because in DNA hybridization, we're actually going to get 2 different sets of DNA and look at how similar the 2 sets of DNA are. Whereas in PCR, we're just having one, we know what this set of DNA is, and we're just looking to basically make a ton of copies of this DNA. So that's kind of the difference between those 2. They do use similar methods in that they are using heat to turn them into single strands. But then basically after that step, they're going different directions. Whereas in DNA hybridization, we're going to put the 2 different strands together and see how much they overlap. And in PCR, when we put the single strands together, we know that they match. We know that these are the same. Sorry, that is like a really terrible color. It's a known match though. So that's kind of the difference between those 2. But that first step of PCR and DNA hybridization is the same. Yes, Michaela, you could definitely add like a mutation and then basically amplify that. Yep, and if you, as long as you basically do kind of like at the beginning from the DNA cloning, where you're putting it in the right direction, it's got a primer or a promoter, that's going to work just fine. All right. So here is kind of a practice question from earlier. Sorry, this maybe should have been a little bit earlier. Oh, do we want to do practice question? Actually, let me review this really quick. So kind of somebody asked me to explain what I meant here by known match. So I was just talking about the difference. Let me see if I can zoom out and maybe put them both here. So we can look at DNA hybridization. And what I was trying to say here is that in DNA hybridization, we've got, if this is like cell A and this is cell B, this is cell A, this is cell B, right? So when we're putting the different single strands together in our container or vial or beaker, whatever you're putting it in, we know that these are different. Right? They came from two different cells, two different organisms, two different zones. We know that they're not, or, we know that they're from different sources. Whereas in polymerase chain reactions, PCRs, we know that this is the same source. So this is only from one source. Whereas you could have two, maybe you have three, maybe you have four in DNA hybridization. You're just basically using different sources compared to one source. All right, I'm going to zoom back in a little bit. So we'll go on to this practice question. You can feel free to type it in the box if you want. Which of the following is true regarding cDNA as used in gene cloning? Reverse transcriptase is required to make cDNA. cDNA library is the same as a DNA library. cDNA has RNA bases. cDNA is synthesized from single-stranded DNA. Anyone else want to throw their answer in the hat? Okay, let's take a look. So I don't know if Todd has taught any lectures, but he taught most of my lectures when I took Berkeley Review. So I like to use kind of like a process of elimination. I know. All right, so reverse transcriptase is required to make cDNA. When we think about cDNA, right, we're going from mRNA back to that cDNA. We know we're going in the reverse and we're basically transcribing. Reverse transcriptase is true. We need that. cDNA is the same as DNA libraries. No, this is going to be false. Oh, sorry. Maybe I shouldn't have put a check here. We'll go for a T. True. This is going to be false because we know that the cDNA library, right, is telling us what the mRNA transcribed, whereas a DNA library is telling us, oh, sorry, maybe we should have done it this way. A DNA library is telling us the DNA code for some protein, but we're starting with the protein and getting back to the DNA library. So you know that those are not the same. cDNA has RNA bases. Well, we said that this was done via reverse transcriptase, which I'm just going to put as RT here. So we actually know that we've replaced any uracils. So this is going to be with DNA bases. That's why it's called cDNA. And cDNA is synthesized from single-stranded DNA. Well, actually, cDNA comes from mRNA. So it's going to be false. So true. It's going to be A. So you kind of sometimes you just have to understand those big kind of overarching things. Protein to DNA library, right? Or mRNA to cDNA. So those are kind of how you can make those two friends. You could probably come up with an acronym. There are a bajillion acronyms. Your brains are probably exploding with acronyms. So we just got a few more. Any questions on that before we go on? All right, just a few more slides here. So why would we even want to do any of these kind of experiments, tests, examples? Well, basically, we're looking to understand gene expression and gene function. So genes obviously give us products of some form, often proteins. Sometimes they give us rRNA, which is a part of ribosomal RNA. Sometimes they give us tRNA, which is transfer RNA, or snRNA, which is small nuclear RNA. Those are just some of the different things that we might be looking for. So when we manipulate genes, we're looking for a change in these products, or maybe a change in the behavior of a cell. Maybe it's going to be fatal to a cell. That's going to be pretty easy to see if there's a change or not. So when we're looking to see what the function is, maybe we're going to knock out the gene. We're basically just going to like have a restriction enzyme cut it out and then see if the cell changes. Does anything, is it non-functional? Is it no longer able to, complete some duty that it's doing? Maybe it microtubules don't work anymore. And therefore we know that that gene is related to microtubules. So we test it out. We can also use basically reverse genetics. So we'll look at the sequence of the gene that we're looking at. So let's say we have this gene of interest right here, and we're looking at the sequence, and then we can look, right, in a DNA library. We're going to look for similar sequences in a DNA library and see what they do. Look, maybe, oh, look, there's a gene that's basically the same sequence, except maybe two different nucleotides are different, and it is functional in the cell membrane. It really helps with the fluidity of the cell membrane. If we'd knocked out that gene, our cell membrane is no longer fluid. So then maybe this gene is also related to the cell membrane. So those are just kind of some of the big picture ideas about why we would even bother looking at this kind of like gene testing, gene cloning, PCR, all of these different lab techniques. This is basically like our big picture. Why are we doing this? Why are we doing this? Well, we're going to look basically at what it's making and how can we change the function. If you have not already realized, the MCAT loves to kind of drive home over and over again that structure determines function, and kind of part of, I would say, structure is like even down to the DNA structure. So we're always thinking about structure and function and how they interrelate. The last kind of like step of this lecture, or that's included, is what we think of as stem cells. And so this at the top right here is kind of important to remember. I'd star it. But in order to be defined as a stem cells have to have the ability to self-renew, and they have to have a high capacity to differentiate into other cell types. That's considered potency. And so kind of understanding this basis of like the definition or these two qualifications of stem cells, we can think about like where we might find stem cells. You've probably heard like of examples of stem cells or stem cell therapies. And so you kind of, if you think about that, if they're self-renewing and they have a high capacity to differentiate, they could be really helpful. So if we think about where we all came from, most of us, if not all of us, came from a zygote, which is what we call the sperm meeting the egg. I'm not sure if you guys have had repro yet, but some of these words will seem more familiar after that. Basically, the sperm meets the egg, they're going to start dividing. Eventually, we get something called a blastocyst, which I'm not sure if you've seen it, but often kind of is represented like this. That's like a little blastocyst. And within this is this cluster of cells, which is called the inner cell mass, the ICM. And this inner cell mass is eventually going to become the embryo, which eventually becomes us. So we know that within this inner cell mass, there are definitely going to be stem cells that have a high capacity to differentiate into other stem cells types. So what's important to note is that within this mass are both pluripotent, which you'll see right here, and totipotent stem cells. And there is an important differentiation between those two types of stem cells. So pluri, probably by the name you've guessed, could become many different cell types. Pluri means many. Potent is that like differentiation ability. Totipotent, does anybody know what toti is like the prefix for, or like what the Latin meaning of toti is? You might have heard of another word similar, Total. total. And that is a good way to remember it, because if you think of totipotent, it's the total thing. So totipotent cells can become an entire organism. So within this inner cell mass, obviously, we don't stay as a mass of cells the entire time. Well, we do, but we don't stay within this blastocyst form. We become a full organism. We become a full human. So there are totipotent cells within that inner cell mass, but there are also pluripotent cells. This is one of the few places that totipotent cells are found. So if you ever are seeing like where are totipotent cells found, think always back to the inner cell mass and that super early embryonic development, because that is one of the rare places that totipotent cells are found. Pluripotent cells, while still rare, are slightly more common, and they might be kind of found in other places. So that's kind of the difference between those two. You might also hear this inner cell mass and the kind of combining of pluripotent and totipotent cells as embryonic stem cells. So that's just kind of giving you another like vocab of just like making those similar. In our body, we also have somatic cells, stem cells. You might have heard that I think it's every day you lose some ridiculous amount of skin cells. I want to say it's like 40%. That could be totally wrong. Don't quote me on that. But your skin is obviously shedding, particularly if you have dry skin. You can see it. You know it's everywhere. But obviously, we don't just start bleeding over time. It's not like our skin never repairs itself. So there has to be something within our skin that's actually regenerating and rebuilding. And this is a form of a somatic stem cell. And those stem cells in this particular example are actually epidermal stem cells. So kind of along the base layer of the epidermis. If you've seen any like cross sections of skin, you maybe have seen kind of like the epidermal layers. You know that there are tons of different cells within skin, but the epidermis is our kind of most external layer. And there are epidermal stem cells, which are able to basically regenerate themselves and also create new skin cells, new epidermal cells. And so because the epidermal stem cells can only create epidermal cells, they're considered unipotent, meaning they can only make one thing. They just make epidermis. That's their job. Compared to when we think about bone marrow, what typically are we making in the bone marrow? Does anyone know? Blood, definitely. And maybe if you've been to like a really fancy restaurant, sometimes they will serve the bone marrow, and you can like literally suck the bone marrow out of a bone of some animal. Sorry if you are vegetarian or vegan and that's disturbing to you. It's personally not up my alley, but apparently it's really good. But in the bone marrow, we make blood. We make red blood cells. We make white blood cells. There's a lot going on there. So when we have some hematopoietic bone marrow stem cells, those are going to be able to differentiate into many different types of eventual cells, lymphocytes. Maybe you have like, maybe potentially, although most of them are made. Sorry, that was kind of confusing. They might be able to turn into lymphocytes, white blood cells, red blood cells, potentially like heme groups, all different sorts of things. And so those bone marrow stem cells are considered multipotent because they can become multiple different things. Sometimes you also see this as oligopotent. And if you think back to like oligosaccharides, it's just that word for like few is oligo. So if you see oligopotent or multipotent, know that those are the same things. They become a few things. But as I said, remember the inner cell mass is where we have our pluripotent and totipotent. Those are considered the rarer ones or more rare. And then kind of once we're differentiated, we've got somatic, which are either unipotent or multipotent. We're going to go over just the two different ways that they divide. I think then there's one more question and then maybe we're done. So hopefully that's good. But basically stem cells can divide under two different processes. One is called obligate asymmetric replication. So you have your stem cell, which I'm just going to call SC. It is going to divide into basically a mama cell and a daughter cell. And in this mother cell, this basically becomes the new stem cell. And this daughter cell will undergo different differentiation this is my very simplified version of differentiation, to become that differentiated cell. Different. We're just gonna go with different. So this is obligate asymmetric. As you can tell from the name, asymmetric, meaning these are not the same. It's not just mitosis that's going on, because obviously that would result in two of the same products. So it's obligate asymmetric replication is a really good way to think about this. And this is often called the mother, like mom and daughter, which you might remember from like mitosis and meiosis when we talk about daughter cells. Compared to stochastic, don't know where that word came from, but that is when a stem cell, which we're going to call here, I'll just write that out for you as well, in case you're taking a screenshot, is going to become just two daughter cells. But then you think, well, the idea of a stem cell is that it's able to self-regenerate. So in this case, there is a nearby stem cell who somehow receives like a little signal that says, hey, I just differentiated into two daughter cells. So can you help a sister out? And it is going to undergo mitosis to produce two stem cells. So this is kind of like a two-part regeneration or differentiation, where we have the first part is creating these two daughter cells. And then in the second part, we're creating the two stem cells. So these are two different ways that stem cells can undergo division. And it's just kind of helpful to maybe remember those in case they pop up in a question or you see these terms thrown around, and you can just identify those with stem cell division or differentiation, or kind of those two main, I guess, like the main like rules for stem cells is that they have to be able to regenerate. Did I spell that right? Stem cells have to regenerate and they have to be able to differentiate. Those are our like two rules for stem cells. These are the two ways in which they can achieve that. Just something nice to kind of balance. I don't know if anyone wants to take a screenshot. I should have said that at the beginning. I'm totally fine with you taking screenshots. This is just all random things. It doesn't really do much for my street cred about me handwriting or anything like that. I'll move on in 5. Okay, good question, Alexandra. This is like a 2-step process. So these steps are required to be done together or like done in sync. So the first step would be that this original stem cell differentiates, and this is a different stem cell. So maybe I should have done it in a different color. This is like a neighbor stem cell that receives the signal about this differentiation into the two daughter cells, and then basically like makes up for that lack of the stem cell and produces two. So that no matter what, we still have this original, basically, like if we claim this was we'll see. You know that this is not the way it works, but I'm using the colors to help us. It would be like this kind of original stem cell is ending up over here, and this is the other original stem cell ending up over here. So we still end up at the end of the day with our two stem cells. So we don't want to like lose any stem cells. Otherwise that could be damaging. No, so this stem cell is just a totally different stem cell over here. The second step stem cell is just like a neighboring, this is like a neighbor. It's nearby, it gets the signal that this other stem cell has differentiated, and therefore it needs to make up for that loss of that stem cell by basically copying itself so that they can still have two stem cells and continue going on. Their whole goal is to like keep the stem cells alive, be able to regenerate, and keep that quota up. And that's important kind of in what we'll go into next for like the biomedical applications. If we thought about it, let's think about our skin in that example. Maybe this will kind of help. If we thought about those epidermal stem cells, if they get like they figure out that they need to divide, if they only became skin cells here, and then this moved on to become whatever differentiated cell it needed to be, right? I can only do that once. Then I had no longer have any more stem cells. So it's helpful for the stem cell to be able to regenerate this like part 2 over here, so that I can regenerate the stem cell again, so that I can still have that process going. Correct. Compared to the obligate asymmetric replication, where this is the original stem cell, and now my mother's cell, or my mom's stem cell up here, is going to be basically the same stem cell, right? So yeah, maybe this is confusing the new. This is going to be the same as the original, basically. It's a full copy of itself, and the daughter is going to get differentiated. But this stem cell up here can now do the same. It can reproduce itself, and it can reproduce another daughter. And it can reproduce itself, and it can reproduce another daughter. So that it can make sure that the stem cell is constantly regenerating. There's always a backup. It's kind of like we need that base to keep going. We have to make sure that stem cell can regenerate itself. That's part of like its like dogma of who a stem cell is. It regenerates and it's able to differentiate. And these are just kind of the 2 processes that it does it. So the obligate is a 1 step. Maybe that is helpful to remember. Obligate is 1 step, and stochastic, which has a T, stochastic is 2 step. Not sure if that's helpful. Just kind of popped into my mind, but we can kind of come back to this if this is still feeling not super certain, and I can try to explain it a different way. All right. Last little part that we'll do, I think, before one question. So the biomedical applications, you might have heard of this. If anybody's working in regenerative medicine, this is definitely something that is of interest. But what we can do is we can make what's called an induced pluripotent stem cell. You might see this as IPS, or I've seen it more commonly as iPSC. And basically what you do is you take some differentiated cell up here. So this is just happy little cell, and it's living its normal life, doing its normal thing. And we can insert into it a gene that basically reverses this cell, so that now, oh, that lime green is terrible. I'm so sorry. Now the cell, I'll do a different color actually, is like not sure of itself. It basically has gone back to now a pluripotent self. But it obviously didn't start that way, right? It was differentiated over here. This is a differentiated cell. This is like a mature cell, one might say. It has a job, it has a function, it knows what it's doing. But when we introduce this new gene, or this gene that's going to help reverse it, it now becomes a pluripotent cell. It has a whole host of things ahead of it. Maybe you kind of feel like an induced pluripotent cell. You have, gone to college and you decided to be a musician, you have done your whole musical thing, and then somewhere, somehow, someone brainwashes you, will claim that that gene is brainwashing you. And now you're back to the beginning. You have no idea what you want to do with your life. Do you become a doctor? Do you become an engineer? Do you become a musician? Do you become a teacher? That's basically what we're doing to this cell. We're like flipping it on its head and sending it back to this pluripotent state where it could become really kind of any kind of like cell. Someone asked, is there a good way to remember the difference between multipotent and pluripotent? Is it multipotent and pluri, or multi and toti? Maybe qualify for that for me. But difference between multi and pluripotent, I would say multipotent is typically when we are looking at like a mature cell or like cells that potentially are able to become different things within a family. So multipotent, when we think of the blood cells, hematopoietic cells, we're sticking within that family. Whereas pluripotent, this could become a blood cell, it could become a nerve cell, it could become a skin cell or a mesenchymal cell. So multipotent, is within a family, whereas pluripotent is just kind of any. I don't really have a good way to remember the difference. If anybody does, feel free to chime in. I think some of them are just like practice. Pluri is plural, I guess. So maybe that's like more families. I don't know. You could maybe look it up online too. There's probably some answers on there. So as I said, these are used in regenerative medicine. If you've ever heard of that field of medicine, I've been to a few conferences on regenerative medicine, and it's really cool. They have basically taken someone's cells, often like a combination of cells. So it can be blood cells, it'll be like skin cells, they'll just get like kind of a ton of different cells. They will introduce these genes to kind of like reverse the differentiation, basically like make them immature again, produce these pluripotent stem cells, and then they will build skin grafts with them. I went to a conference where they were able to talk about building someone had like really bad ureter damage from like a genetic defect. They rebuilt a ureter for this person. They had like a mesh kind of like structure that the cells could grow on, but they were able to like induce them to be able to like rebuild the structure of a ureter. They're doing it now with organs, kidneys. I think I heard that they're starting to do like a heart. And so regenerative medicine is a really cool opportunity because the idea would be that if this is a mature cell from me, this now becomes a pluripotent cell that technically is from me. And therefore, if this becomes maybe like a kidney, when it gets transplanted back inside of me, I already know it. It's already self. It has all those self markers. And so therefore there's a smaller chance of rejection. And it could be really beneficial in like self repair. If I had gotten a ton of burns and I needed skin grafts, a big challenge with skin grafts is rejection. And so if these cells are kind of made from me, there's a lower chance of rejection. Of course, often in an emergency, we don't have time to like grow this in a lab. So, there's definitely some limits, but it's really cool, really interesting avenue of medicine. One last one that sometimes they ask about is cord blood. You may have heard of it. The thing to know about cord blood is that it basically contains a lot of multipotent, which we said are like the family. So could make blood cells or could make bone cells or could make nerve cells, and then pluripotent, which can make technically any cell, but not an organism. So that's why people might bank that in case that's becoming really popular. So those are kind of that. I have one last question. If you want to answer it, you can answer it on your own time as well, which is the least potent stem cell. Feel free to chat it in the box. All right. Anyone else? Anyone want to explain why? You can feel free to take yourself off mute and explain why you think it's C or maybe a different answer.
I said C because I think you said earlier that the epidermal stem cells can only become one other or can only become their own kind of cell, whereas I think the other ones can become more than one. So it would seem the least potent to me, but I'm not sure if that's right.
Excellent. That is exactly right. When we think about these things, we're looking at like potency. Remember that potency is the ability to differentiate. I'm just going to leave it as differentiate. So a bone marrow stem cell, remember that can make all blood cells. And we know there are many different types of cells that make up our blood. So this is what we would consider multipotent. When we think of embryonic stem cells, we have two different types of stem cells in there. We have both pluripotent and totipotent. That can make a whole human, right? It can also make the nervous system, the digestive system, the integumentary system, all of those things. Whereas an epidermal stem cell can only make epidermal cells. So therefore, it's called unipotent. And this was almost exactly a question that I had, I think, on a practice test from the AAMC. So this is like definitely right up their alley in terms of that. I think that's my last slide. Yeah, that is my last slide. So definitely feel free to ask any kind of end questions. If you did express like maybe some interest in like a review cell, if you don't mind either emailing me what topics might be helpful to review or just throwing it in the chat box as well. I'll kind of jot those down. It seems like Central Dogma was requested, as well as just maybe some like Gen Bio. But happy to kind of like maybe chat with, can't remember his name currently, but Josh is the word I'm looking for. Sorry, don't tell him that. But yeah, chat with Josh to see if we can maybe get some reviews. Somebody asked, what's the best biology resource? I really enjoyed the Berkeley Review books. I thought like doing as many practice questions as you can. Yes, when you get to a practice question and you're like, cool, I have no idea what this is. This is absolutely crazy. Maybe I'll actually stop screen sharing so you can like see my face if that feels less weird. I thought it was kind of comfortable, or like it was more helpful for me to review, go through practice questions, not like a practice test. Like I'm talking about like in the chapters or like in the big reviews in each of the books. Kind of going through, even if you guess and you get something right, being able to like mark on there, like I guessed this, I actually need to go back and review this topic. And then being able to review, I also am a big Khan Academy person. Some people don't like it. I really enjoy like visual learning and being able to watch someone draw out a sequence or just like explain it in a different way. And they partnered with AAMC to actually like create free MCAT materials. So most of the stuff that's up there is like exactly basically what the AAMC wants you to know. I did also have, if I remember correctly, I think they were from Kaplan. My friend gave them to me that were like these kind of like plastic cards, or like these big, like, I guess they were paper sized, of like just random things about biology. And sometimes that's nicer once you're getting close. I don't know if anybody's close to taking their MCAT, but you don't necessarily want to be doing like heavy lifting topics. You're just kind of looking for those basic refreshers. You could also make them yourself. But I think really any way that you can practice, if there's like question a day type websites, I really loved doing those, just to kind of like see what's out there, what's going on. Yeah, in terms of topics that I'm okay with reviewing, I would say I feel like pretty confident in the biology wheelhouse. I also really enjoyed sociology and psychology. I know those are a lot of terms to remember, so sometimes it just gets draining on your brain. Cars, you guys are in good hands with Patricio. And if you haven't checked it out, I felt like Jack Weston was a really good like daily. I did not pay for it. I just want to make that very clear. But I just did like the daily ones that were free. I just thought it was a good practice. It was a really nice way to like sneak in a 15-minute study session in a way that actually felt really practical, and like helping read stuff was good. Chemistry, I really don't enjoy. So I'm sorry if you're really struggling in chem and organic chemistry. It's not my friend. But yeah, I would say those are the topics. Definitely good. If there are any other like topics to review or like things that you would want to review, just throw it in the chat so I can kind of catch those. Let me just see if I missed any other questions. What are the highest yield straight biology topics? I heard it was enzyme kinetics, amino acids for biochem, and for biology, you need to know cycles and glycolysis. I would say that's fairly accurate. I think though, honestly, like if you try to think about, this is not what you want to hear, but I'm going to tell you anyway. If you try to think about the biology section as like what's going to be the highest yield, you could spend all your time studying each amino acid, knowing the pKa's of the amino acid, and you might get one question on an amino acid. And like, that's what it is on the day. So it does really, I think, depend. I would obviously try to know the different amino acids, maybe like what their families are, polar, non-polar, just different things like that. And generally, like pKa is in this ballpark. But even then, like, if I had to drop one thing, maybe for me it is the pKa's. That's just not going to go with me. And I'm really going to like dive deep somewhere else. You kind of have to know what your personal balance is. So that's really like kind of a personal go with your gut kind of thing, which I know is like really hard. And, AAMC wants you to know everything at a like super detailed level. So it can kind of feel tough in that sense, but somebody asked, should we know about glycolysis and electron transport chain? I would say those are definitely going to be like, if you can feel confident on those, you might get to something that is considered like a tougher question. But if you feel really confident in those details, you're going to be able to tackle that question with a lot more confidence. So I would kind of like think about what's the best way for you to like understand that. For me, I recorded myself saying the steps of glycolysis and what each product was and what enzymes might be involved or what was like a reversible or irreversible step. I recorded that as like a little audio recording on my phone. And then anytime I was walking somewhere, I would just like throw that on and basically make that be like muscle memory to me, because that helped me feel a lot more confident about glycolysis. Krebs cycle, definitely you kind of want to know there's like some inappropriate acronym. Just if that works for you, that works for me. I can't remember it off the top of my head. But just to kind of like know what products you're going to make, I think is more that way. Sometimes they'll ask you like, you're given this, which is a relative of this. And so like oxaloacetate, and where does that come in? Well, it's Krebs cycle. You could probably like figure out how many steps away. I would say those are probably like, if you want to call those heavy hitters or highest yield compared to pentose phosphate pathway and urea cycle, but you don't want to totally ignore those. You kind of have to like keep that balance of knowing what you personally are going to like really get confident in. And sometimes that's what you're not confident in and what you're just going to like keep fresh in the back of your mind. Like urea cycle for me, like I was like, yeah, I'm fairly okay with that. But, a couple days before my MCAT, I like reread over those notes. I wasn't doing any avid studying on the urea cycle, but I was just kind of keeping those things fresh and relevant and yeah, not really worrying like too much about details once you get close enough. I don't know if anybody has it coming up soon. Is the MCAT even on right now? These are things that I do not know. I think maybe there's like those weird breaks that it has. So yeah, any other like general questions? I can go back to the beginning actually. Thanks for all the congrats. I am going to the University of Queensland. Although it does not sound like it, I actually am Australian. So I get to go back under like citizen rules. So University of Queensland also has a program that semi-partners with Tulane. It's called Ochsner, in case you're interested, where you do your first 2 years in Queensland, which is in Brisbane in Australia. It's really tropical and really nice and like beachy. And you do your first 2 years of training at UQ, and then you come back to the Ochsner medical system, which is in rural Louisiana, although most of Louisiana is relatively rural. So that's kind of that program. If you're interested in that, if you're not an Australian citizen, they also have international applications. If that is something you are interested in, someone asked what made me go to school and choose school in Australia. Honestly, if you have not started applying already, medical school is like really tough to get into. So I applied a few times. to medical school in the US, I interviewed a bunch and just kept getting waitlisted. So I decided to take the GAMSAT, which is the Australian like medical school entrance exam, and took that. And the interview process is like really simple. Applied and got in. A big bonus is that it's about a sixth of the cost that it is here. So that is really, really nice to not like get into $200,000 of debt. And UQ, University of Queensland, has a really big international program, like I mentioned. So it's really easy to come back to the US and practice. But maybe I want to practice there. So it's 4 years, just like the US, 2 years mostly of like theory and kind of like more the classwork side of things, and then 2 years of clinicals. And then you can do step 1, step 2 while you're there, come back for residency. So yeah, it's a pretty good system. Also, if you're hanging around and you're like waiting to see if there's any more class content, there is not. So feel free to jump off. Sorry, I should have said that earlier. If you want to throw in any other topics, you can send them directly to me or email them to me. I'll throw my email in the chat again. You can also let me know if you're interested in like other topics to review or other questions. I can try my best to answer them. But my office hours are 9 a.m. to 11 a.m. on Tuesdays, Pacific time. But if that time doesn't work for you, I know probably some of you are actually like currently in class, maybe you're a junior or senior or you're a graduate student or whatever. You can email me. We could try to set up a different time. I think that's going to be until the end of December. Josh has not like fully kind of told me. If we need to have another day, I'm happy to add another day. We can kind of go from there to see what's best. Just feel free to email me and do that. I see that. Let me actually write a few of these down. So if somebody asked for like lac-operons, motors, somebody asked for central dogma, muscle physio. What else have we got? Genetics, metabolism. Sorry, I'm just scrolling down. I don't want to close our chat before we get this. Sorry, I see 2 new messages, but I'll just check this first. Okay, metabolism, central dogma, genetics. It looks like then muscle physio, glycolysis, endo, maybe. Okay, have you had any endo or muscle physio with Spencer? I don't know if he's still doing physiology this semester. Yes. Okay. So that's good. So maybe I'll touch base with Spencer just to see if I can look at some of his slides. They might still be the same as when I took it. Just to see like how he kind of explained it. Sometimes that's helpful so that I don't explain it exactly the same way, which I know is tricky. Also, if you have any feedback for me about today, I know that I spoke too quickly, so I will definitely try to be more conscious of slowing down. But if there's anything else that you did like or you didn't like, you hated the practice questions, you loved the practice questions, you need more pauses, just let me know so I can work that into next week for cell bio and hopefully into some of these reviews. I know that maybe some of you are feeling a little just like stressed with the end of the year coming on. You might be in finals or also like other things, or you've taken practice tests. So yeah, just let me know. I'm happy to hang out for the next 30 minutes and answer any questions that are going on. So yeah, I know this one was kind of tough with like all of the jargon. I think that's definitely a limitation of like these kind of molecular biotechnology type lectures. So I totally agree with that. And it sounds like maybe if there wasn't kind of a central dogma lecture or like some of this, just like a bio refresh almost, that would definitely be a little bit more confusing. So I appreciate that feedback and definitely will take that into consideration for next time. Definitely let me know if there's any other questions. If you have homework questions, you can feel free to, send them in. It definitely is a balance of like how much you can cover, and I definitely want to be respectful of your time. I was working when I took this class, so I was like really down to the minute, and anytime it even went like 5 minutes over, it was really stressful for me. So I definitely can appreciate if that is a similar spot that you're in. So I'm going to stay online for the next 30 minutes in case, oh my gosh, you've gone 2.5 hours over. I would be furious, is probably how I would describe that. I'm really sorry that happened. That should not be happening. Yeah, that's tough. But yeah, happy to chat. Happy to like if you just need to socialize for a few minutes, also happy to facilitate that. Yeah, definitely feel free to email me. We can figure something out. So I will see you guys next Thursday, same time, 2 to 4, for I think it's like considered just like cell bio. But we're kind of going to go over all of those basic cell bio things. I'm going to try to find a few more practice questions for us and we'll go from there. So have a great weekend. It is Thursday. Definitely get some good studying in, get some good rest in, and let me know how I can help you. Thanks, Jonathan. Thanks, Eileen. See you next week.